So everybody, welcome here tonight. Before we start on neuroplasticity, I just want to reach out to my good friend Tracy Day and her immensely talented husband Brian Green and say how lucky we all are that they took this on 11 years ago and have dedicated so much of their life in the cause of expanding science to the masses. So let's just give Tracy and Brian a small shout of appreciation. So we're here to talk tonight about neuroplasticity. So what actually is neuroplasticity? So neuroplasticity is the ability of the brain to adapt to changing circumstances, to when the environment changes, the ability of our brain to, in a nutshell, learn. But there's another side of the coin. Just as we need to be able to learn and adapt, we also need stability. The opposite of plasticity, neuroplasticity, is brain stability. And so, in fact, that balance is what we're going to focus on a lot tonight. So you think about it, when I do something as simple as this, the simple act of walking, we don't think about it. It's reflex, it's second nature. We all know, we look at it, it's so simple. But in fact, walking is an unbelievably complicated orchestra, symphony of movements, balance, coordination, all together being integrated by our brain. And we don't think that's hard at all because we don't remember how hard it was to learn as a young toddler. But if you've ever seen a patient try to recover from a stroke, or when I see my patients after complicated brain tumor surgery have to relearn to walk, you realize it is actually an incredibly difficult and complicated proposition. So in fact, we as humans, the brain, we need stability and we also need plasticity. We have to be able to learn, but we have to also have things that are hardwired, that are just fixed over time. And so that really is what we're going to talk about tonight, the balance between those two. So as an example more of the way evolution has worked, let's compare. Let's compare the human brain, which is actually the master of neuroplasticity in the world, to the loggerhead turtle, which is actually the master of stability. Our human, newborn Augie Nelson's current abilities are somewhat limited. He can cry, sleep, eat, and hiccup. The turtle, also newly hatched, can crawl, separate from her siblings, and begin a vast transatlantic migration, alone, without parents, or learning. Her brain has already formed every connection she'll need for the great journey ahead. 7 months later, Augie is learning to sit up. In a year or so, he'll be walking. Another decade, he'll be walking to school by himself. By that time, the turtle will have completed a solo 9,000-mile circumnavigation of the Sargasso Sea and returned to the shores where she was born to build her own nest. The turtle's brain was designed to masterfully navigate the ocean, but Augie's human brain was designed to adapt to any environment. Play any instrument, calculate the existence of a space-time singularity, navigate the Atlantic by himself if he wants. Everything he needs to learn these things is already there.
Wow!
We're born with as many neurons and as many connections as you're going to need. They can go from any spot of the brain to every spot with as many connections as possible.
And that's a brilliant design by Mother Nature because it gives us this incredible amount of possibilities for the future.
But there's a catch. That gift of possibility doesn't last forever. Like childhood itself, it comes to an end. For each skill, we get a golden period when learning is easy.
Periods in development when we have a heightened plasticity for shaping neural circuitry, and we refer to those as critical periods.
Learning to play the guitar as a child is like opening a door. And as a child, we have lots and lots of doors.
During this time, our neural circuitry gets fine-tuned.
Our brain learns about which connections are going to be important through a use it or lose it principle.
Certain connections become reinforced and sustained and enhanced at the cost of others.
Those connections that aren't being activated because they're learned not to be necessary get pruned away.
They actually die off, and the open doors begin to close.
As an adult, if you've never played, you've lost that door.
So right now, Augie can learn to speak any human language perfectly
Kalau kamu tidak belajar bahasa Indonesia
without an accent. But when he's an adult, he will have lost that ability. If he doesn't begin learning the violin before he's about 7 years old, he'll probably never play Carnegie Hall.
If you never had the connections to begin with, you can try as hard as you can. You're never going to get there.
Play.
But what if we could change that?
Play.
What if we could tweak our brains to learn as easily as a child?
Wonderful. Let's do it again.
It's called the Holy Grail of neuroscience, and it is tantalizingly close.
We can, to some degree, do the impossible and reopen critical periods, as it were, allow changes to happen later on in ways that we didn't anticipate would be possible.
Scientists are learning how we learn,
And then play the fourth finger.
and that may soon give us a chance to reopen those doors.
Yes.
So we have an incredibly exciting and fun topic to discuss tonight, and we've got 3 of the most amazing people in the world to help us through this conversation tonight. Our first participant was the first woman to chair the Department of Neurobiology at Harvard Medical School. Today, she's professor of biology and neurobiology at Stanford University. She's the winner of innumerable scientific prizes, including the Gruber Prize, the Kavli Prize, the Harvey Prize. Please welcome one of the world's foremost neuroscientists, Carla Shatz. Our next participant is Associate Professor of Psychology at Columbia University, Director of the Developmental Affective Neuroscience Laboratory, a recipient of the National Institute of Mental Health Brains Award. Please welcome Nim Tottenham. And our third participant this evening is Professor of Neurology at Harvard Medical School. An MD PhD, he says his research falls in the area between the brain and the mind. He's a world leader in the use of transcranial magnetic stimulation to study the brain. Please welcome Alvaro Pascual-Leone. So welcome, everybody. So, Carla, let's start with the brain itself. We're going to be talking about learning, plasticity, how the brain learns, how skills become habit. But what do these things actually look like in the brain structurally?
Well, we saw already some nice photographs in the introduction, if you remember, of these black things. And structurally, the brain is composed of neurons and support cells. And the neurons communicate with each other through electrical signaling, chemical electrical signaling. And the most important aspect of that is that these communications are transferred across structures known as synapses. And that's where learning happens, that's where memories are stored. And as we were talking about in the video, that's where this pruning process goes on, this use it or lose it process goes on during development. So in fact, the structure of the brain is just, the brain is packed with these neurons, packed with these synapses, and also with very long connections. And some of those you can actually see in this beautiful graphic that's being displayed now. So the brain connections link various parts of the structure to each other, and those are the beautiful colors that look like U's and so on, these pathways. But those are made up of hundreds and thousands of connections from individual neurons. And I used to joke there are more neurons in the brain than stars in the universe, but in fact, an astronomer corrected me and said that wasn't true.
So when you look at an amazing picture like that, we're looking really at the axons, the wiring.
The wiring.
So is the brain fundamentally unwired at the start?
So the brain is not fundamentally unwired at the start, but it is amazing. If you just take the visual system, for example, the eye is not connected to the brain to start with. And the nerve cells in the eye have to grow their connections along pathways. and select the right part of the brain, namely the visual part of the brain, not the auditory part of the brain or the motor part of the brain. So the wiring from the very beginning is very highly organized, and the formation of these long tracks or pathways is dictated by very strict cues, like pathways with, or even like roads with street signs on them, so that these growing connections or the growing axons follow these pathways. But then the amazing thing is, once the connections form between these long, distant regions of the brain, then there's this period of overproduction and pruning that we're going to talk about, a period of extensive plasticity where the outcome is really predicted by the use of the circuit itself and the experience itself. So there's both directed kind of hardwiring early, followed by this remodeling plasticity that happens later and really persists to some extent throughout life.
So Nim, building on that, if we think about it, if you're building a house, you're not going to lay copper wires all over the place and then pull out the ones that don't seem like they're working well. But that seems to be what we're talking about. And it seems pretty inefficient. And the brain is 2% of the body weight, but yet gets 20% of the blood flow and 25% of the glucose. So why is the brain doing it that way?
Yeah. So on face value, there's so much about brain development that seems inefficient. But actually, it's that seemingly inefficient path that actually gives rise to the incredible complexity of the brain, especially in the case of the human. So when we're developing, what our brain does is first goes through this period of overproducing our neurons and synapses. And then through experience, through learning what's important in the environment, what synapses are getting activated, those get to stay and everything else gets snipped away because it's inefficient to keep all of those synapses. So why would we go through this entire process of overproducing and then pulling back? That's really at the core of what the function of childhood is in the human. So if you think about humans as a species, we're amazing in many ways. We can live anywhere on the planet, we can speak any language, we can eat a number of foods and so on and live very well. And some of the reason why we can do that is because of this long period of brain development that we have. So the brain overproduces. It's like throwing a big fishing net out into the ocean because you're not sure which house you're going to end up in. You're not sure what language people are going to be speaking to you. So you're ready. You've got all your bases covered. And then depending on what signals are coming into the brain learns, okay, I'm going to keep these synapses, but I'm going to get rid of these. So if I'm growing up in a Japanese-speaking household, I'm going to hang on to the synapses that help me understand Japanese, but get rid of the synapses that support other languages because that's really efficient. So it really is this developmental period, a really long one in humans, that gives rise to our incredible adaptation to our particular environments.
So Alvaro, is this a new concept? And we all think everything's been figured out in neuroscience in the last 20 years, but this need to balance stability and plasticity. Tell us a little more about that, about the history of that conceptually.
I think there's lots of new developments, but many of the ideas are not fundamentally new. We have the tools to actually test the concepts, but the first person to speak about plasticity and stability as a term and applied to behavior, to human behavior, was William James, the psychologist William James, in the 1890s. He wrote about plasticity as being that property of a substance, of an organ, presumably of the brain, that enabled us as humans to do certain behaviors and become really good at it. He was talking about behavior as a habit, not about the brain structure. And he said, the amazing thing, it has a property of giving it to influence, but not giving in all at once. You want to have some plasticity, but not too much plasticity. You want the right kind of plasticity, the right amount of plasticity. And presumably it was Ramon y Cajal, then not much longer, who started to look into where does that actually happen in the brain and describe the synapses and the changes in the connectivity and the change of how efficacious those connections were and making new ones as the substrate for that plasticity that William James was talking about. So the ideas are, 100 years old. And yet when I was studying medicine, I was told, things don't change in the brain, and when you get old, well, tough, And I thought, God, I hope we can do something about it, because I hope to get there one day. And I think we start to all know, we've always known, as an old dog, you can still learn new tricks, and so it was against the experience of everybody that it is possible to learn new stuff. So how does that gel together?
Great. So Nim, let's go back to this. We were talking about this concept of a critical period. And we all know the old adage that children learn language a lot more easily than adults. But what are some of the other examples of critical periods that you see in your work with children?
So for a while, people were thinking that there is a critical period of human brain development. That was a language that people used. And then it became clear that critical periods are really a property of a developing neural circuit. And so if that's true, then maybe it's appropriate to think of each neural circuit that we have as undergoing its own critical period. That moment, the metaphor that people often use for a critical period is that it's a window of opportunity opening up when the environment can really have its biggest impact on the nature of future functioning for that system. If it's true that every neural circuit has its own critical period, then maybe we can sort of map out the timing across the brain. And so in general, what people find is that the brain develops in this kind of backward C-shaped nature, where regions of the brain that are low and back tend to develop earlier, and then the development kind of curls around in waves. And so what you often see are, as you can see on this figure, that often sensory systems develop earlier, experience their critical periods, followed by motor systems and language systems, and finally more of these higher cognitive functions that particularly in the human we spend so much time thinking about. So things like our emotion processing or our cognition, academic performance, and so on. So that hierarchical structure really makes sense when you think about what the needs of the developing infant, child, or adolescent are going to be.
So the reason why the brain does it that way is because as we're older, we need these higher functions?
The way that I think about it is that the more complex the functions, the higher the functions, the more lower level information they're going to need in order to be informed about the environment.
And what about outside of vision and movement and language? What about things like social skills, emotional skills?
I think one of the really exciting things that is being discovered is that even these more complex, higher level skills may also undergo periods of heightened plasticity. Maybe we wouldn't want to call them critical periods in the strictest sense, but maybe more sensitive periods, that the boundaries, the window opening and closing, the boundaries may be a little more forgiving and stretched out over time. So rather than a critical period opening up for a few weeks at a time, when we're talking about these higher level functions, really we're looking at a much more protracted time window.
Maybe we should also clarify a little bit this concept of opening and closing, in the sense that I think, at least for the visual system and for other sensory systems, the notion is that there are these critical times in development when the system has to be used in appropriate ways in order to make the appropriate selection of connections and to function properly. So that's a critical period. And if you think about a window or a door closing, then they don't really slam shut, in the sense that there's ongoing plasticity throughout life, but it seems as if the amount of plasticity that is available, and maybe even the quality, and maybe even the underlying molecular mechanisms for this plasticity, might be altered or diminished as we go through these periods, and maybe the window stays cracked open a little bit.
Yeah, and I'm really glad you said that. I mean, language is a very good example of that clearly we can learn a second language as an adult, but it's going to require a lot more effort and a lot more practice than it would have when we were 2 years old.
Yeah, and I realize this is my dream, is to learn French without an accent.
Right.
It's not going to happen unless...
Well, I can attest to that.
Yeah. Every year that I get older, I'm very, very thankful to know that the door stays ajar. But it does bring up the point that in the video we saw a baby turtle that really doesn't have a childhood. It jumps in the ocean and takes off, whereas humans, we have this extended childhood period where all this has to happen, and you could argue that because we have the most to go through experientially, that we have the longest childhood.
Mm-hmm. I think it's another good example of what seems like an inefficient design, right? So if we're going to stay immature for that long, then we need somebody around us who's willing to put in the investment to stick around with us that long. So if you look at this immature period in the human, it's really a long time, right? In most species, this period of immaturity is on the order of weeks, maybe months, and in humans, it's years. And so that's a curious design for Mother Nature to come up with, because it's a huge energy suck for a parent, right? To raise another human being. I don't know what the numbers are, but someone computed how many calories it would take to raise another human being, and it's enormous. So there's got to be a really big payoff. And one of the arguments is that the payoff is that this affords us as a species this incredibly long period of plasticity so that we can do all of the learning that's necessary to become a very complex adult.
But so at the same time, I think what we're learning is that this notion of development, and then you get to some level of maturity, which I'm still hoping for, and then you have a plateau, and at some point things go wrong. If you are unlucky, you start losing it. That this way of thinking about it is probably the wrong way to think about it. That instead we should think of us sort of developing our entire lifespan until we sort of die, and that therefore plasticity, even though it may be through different mechanisms and different efficacies and working on different substrates, but it's still there. It's still there throughout the lifespan, and so it's not a critical period in the sense that plasticity is done and now you can reactivate plasticity. You can open opportunities, but the capacity of change, balancing the stability, is there for the entire lifespan, which is, from a neurologist's point of view, a neurosurgical perspective, a huge opportunity and reason for hope of interventions.
This balance between stability and plasticity is really interesting. If you just think about some of the systems that have to form, then you don't want necessarily plasticity throughout life in a system, let's say like our visual system, where we need to have a stable representation of the world inside our brain. And then we can draw upon that to make computations, to put together, and to have perceptions and so on. So it's really quite interesting. So some systems really need to go through this learning period, but then to become more stable than other systems.
So let's build on that a little, Carla, because the visual system, your mentors at Harvard, Hubel and Wiesel, won the Nobel Prize in 1981 for their fundamental work on really the first insights into all this concept in the visual system. So help us understand that a little more.
Oh, sure. I have to just make a little joke. So this is David Hubel and Torsten Wiesel. And my mother, I used to call them Hubel and Wiesel, and my mother thought it was one person, Hubel and Wiesel, till she met them. But yeah, so these two wonderful scientists explored the visual system and began to try to understand how it is. Well, here's the question: how come we see with one view of the world, even though we have two eyes? So both eyes have it's like you have two cameras. I mean, both eyes bring complete images of the world to the brain. Yet, unless there's pathology, we only see one view of the world. And the answer to this is both in the wiring of the connections and also the fact that the brain has to learn how to use both eyes together. And it does that during a developmental critical period. So the connections from the two eyes begin to be mixed together as the connections move from the periphery, from the retina itself, into the central nervous system. And the first binocular cells are actually built at the back of your brain in your occipital cortex. That's your primary visual cortex. But even though the connections know to grow to the visual part of the brain, they actually don't know how to tune up to make binocular neurons. And one way they do that is to interdigitate the connections between the right eye and the left eye into a series of beautiful right eye, left eye stripes. And you can see that image on the screen now. And you'll see two images here, actually. So there's an image where there are white and black stripes, and those stripes are equal in size.
On the left.
On the left. And then there's another image on the right where there are little teeny black holes in the middle of a sea of white. Now what you're looking at, let's just say on the left, every little white dot is the size of about a synapse, so one of these connections, and you're looking then at literally millions of these connections. What you notice is that they're beautifully organized in these stripes. And when Hubel and Wiesel first discovered this intermixing of the two inputs, which are essential for making the binocular neurons, everybody thought they were hardwired. But they did a very important experiment, and the experimental result is on the right. Let me tell you about it in just one other context, which is really to talk about the mystery of the cataract. So you know perfectly well if, as an adult, you have normal vision your whole life and you get a cataract as an adult, and you lose the vision in your eye because there's a clouding of the lens, then a miracle can happen in that you go to the surgeon and he replaces the lens with a clear lens, and you see beautifully again immediately. And this replacement could happen after maybe 10 years of not being able to see well through your eye. Now, in contrast, a little child who might be born with a congenital cataract or have some other problem with vision in one eye, if that cataract is not corrected immediately, then that child will be permanently blind or have severe vision loss in that eye. So what's the difference? Grandma had a cataract, 10 years worth, corrected, good vision, and then the child maybe has a cataract just for a year, maybe even half a year, and is corrected. The optics of the eye are corrected, the camera works, but the brain can't see. So what's the difference? So Hubel and Wiesel did an experiment where they actually checked the connections between the eye and the brain, and that was shown, if we can maybe have that image up again, that was shown in the right-hand side. The open eye, the white eye's connections take over way more than their fair share of cortical circuitry for vision. And the closed eye has those little piddly, black holes. And this was an amazing demonstration, a very important demonstration, in fact, the first, of the use it or lose it concept in the brain, that the brain connections require use in order to be maintained, and in fact even require use in order to be formed.
So can you go into a little bit how you've been building on that work? And I think you've got a video for us of how the brain does this in real time.
Well, so this really illustrates a developmental critical period that happens after birth. But actually, we were quite interested in knowing whether there are earlier developmental critical periods in the visual system. And what we discovered is that even before babies are born, the eye is sending signals to the brain to start to really shape up these beautiful stripes and these connections in the central pathways. And the signals are the electrical signals that are being sent from the neurons in the retina into the central part of the brain. And they're actually like test patterns. They're testing the connections, and the ones that are appropriate are being maintained and the other ones are being pruned away. So this is the same theme. And the video that you see now is a picture of that signaling process in the eye. So you can just imagine it's like phone calls being placed to the brain really early in development. And every little black dot here in this video is, again, about the size now of a nerve cell, not a synapse. So it's a bigger scale. scale. But you can see that when the cells become black, it means they're placing the phone calls and they're sending their signals to the brain. So what you're seeing here is neighborhoods of nerve cells all placing phone calls together. And this is actually part of another principle of development: cells that fire together wire together. So this is a way that the eye can sort of test to make sure the connections are orderly in the target structure. This is happening, so the brain is really jump-starting vision even before vision is possible, because it's in utero and it's before the rods and the cones have appeared. And now that we know that this happens in the visual system, it's been found that this kind of jump-starting and testing is happening all over the brain during development, very early development.
So, but it's fascinating because you're showing that in the visual system, this is happening with the eyes closed before a baby is born.
And even before vision, so because they're not even seeing
yet. So there's no
yet.
vision. So,
right.
so vision goes, and yet it becomes more rigid once it ends. So that door is more, not closed, but a little more closed than, say, language, where we talked about, you may not be a perfect French accent, but you probably could learn French right now.
Yeah, well, we can discuss that one later, but yeah, I mean, I think that's true, except the other thing I want to emphasize is that the amazing thing is the door is open much earlier than we thought. It's open in the womb, in the developing child, in the developing baby, and which brings the point that anything that interferes with this sort of phoning process can alter the outcome of development, including things like drugs of abuse, for example,
and
which act at synapses.
You've been studying ways to potentially reopen the visual system. So tell us about that a little bit.
Yeah, so well, the question is really, if these windows close, can you open them up at any time? And really, to try to understand how to do that, it's important to know something about the molecular mechanisms. So what are the molecules that are opening and closing these developmental critical periods? And in particular, what are the molecular mechanisms that control the pruning process itself, this selection process? Which synapses should we keep? How does the brain know that certain synapses have been used and they should be strengthened, and other synapses have not been used so much, so we don't need them, so we can actually prune them away? And so we can use animal models to begin to discover those molecules for pruning. And in doing that, in fact, we found a number of candidate molecules that we wanted to test to see if they were important for this pruning process. And so we engineered genetically mice that lacked these molecules to see what would happen to their critical periods and to pruning. And to our great surprise and delight, what we discovered is that if certain of these molecules, when they're not there, permit the persistence of the developmental critical period in the visual system, and in fact, pruning fails to happen. And so this is illustrating 2 points. One is that it is possible to continue, to extend a critical period for a longer period of time. And it also indicates the idea that that brain plasticity itself can be regulated in a very deliberate way. And if we only knew the whole story about the molecular mechanisms, we really could make pills, and I could take a pill as an adult and learn French without an accent.
So that brings up an interesting point, because Alvaro's over there smiling and shaking his head. So as a clinical neurologist...
I've taken the pill.
Exactly.
No, but would you want to take it right away?
So as a clinical neurologist, and you think about that, and we talk about the balance between stability and plasticity, and as you're taking care of patients with strokes and things like that, what do you think about, and then what do you think about in kids as, because everybody's obviously going to say, oh, well, we got to get these into humans. We got to figure out ways to extend these windows for kids and for diseased adults. So let's talk a little bit about the pros and cons of that.
Yeah, so I think it's a really important topic, and it is a double-edged sword, right? To have a great, efficient, plastic brain that is able to get us to learn French or English without an accent, it may be very appealing, but at the same time, to have too much plasticity can come with a cost. If you have not a genetically engineered mouse, but a disease where by design, because of the pathology, the brain is too plastic, is learning too fast, that seems at face value a great thing, right? You're able to acquire skills that go above the average one of us. You can open up a box of matches, have them fall on the ground and say 27 and be right, or 225. You can learn a whole book of names and phone numbers. And we call those savant abilities, and it's sort of cool. And it's certainly a party joke. But it comes at a cost, because what happens to the brain is that normally it yields to influence, to the environmental change, and then we stop, and then we experience a new thing, and that new thing falls onto fertile grounds, ready to learn the new thing. But it doesn't get colored by what we just learned. The risk of having too much plasticity is that the brain is change, and then the change falls onto the change, and a change onto the change onto the change. It's a sort of a messy, noisy system. And we think that, for example, diseases like autism are characterized by excessive, too good a plasticity that leads to a failure of pruning and to a developmental disorder because of an abnormality in plasticity. So to have too much may be a bad thing.
So you've been studying that in autism specifically to try to look at plasticity in the autistic brain versus in the neurotypical non-autistic brain. So what do you find? How do you go about teasing that apart, and what do you find?
So you have to come up with experiments, as it were, to show that people with autism can be better at learning, for example, than individuals that are not autistic. Things like learning a sequence of finger movements that repeat themselves, and you need, by doing it, to figure out the pattern underneath the finger movements. Individuals with autism learn to do that faster than those without autism. But if you have one pattern followed by another, then they break down, because they run into interactions between the two things that they are learning. And at the same time, we want to look in the brain and see what is changing in the brain to allow them to learn these skills faster. And we do that in a sort of similar way that we've learned to do it in animal models or in slices of the brain, using electric stimuli to evoke a response, and then using trains of little stimuli to modify those connections and see for how long they remain modified. We use that using a technique called transcranial magnetic stimulation.
So tell us a little more about that technique and how you're able to use it non-invasively to test something like this, to perturb the brain transiently.
It's a bit like science fiction, still, to me. So you basically put a coil of copper wire over the subject's head.
And you have a video to show us, right?
We have a video, yeah. Then as the current passes through the copper wire coil, it induces a current that goes through the skin and the skull and induces a current in the brain. Maybe we can show it, and there's a little explanation that goes with it. TMS stands for transcranial magnetic stimulation. It is a way of inducing current in the brain, in a specific controlled part of the brain, without having to open up the skin and the skull. And it turns out that when you apply repetitive stimuli, then you're activating a circuit over and over in a controlled pattern, and that changes that circuit. And so that allows us to activate, probe, disrupt, or suppress activity in different parts of the brain, depending on where we target and what parameters of stimulation we apply. And so you can probe the brain with this technique, see the response, apply a little train to modify it, and then what you find in people with autism is that the effect of that modification lasts literally longer than it does in neurotypical subjects, in those without autism.
So people with autism can learn the task better, but when you perturb it, it then takes longer for them to recover from that perturbation. Is that correct?
When you perturb them, the impact of the perturbation is stronger. And that's because of the fact that with the perturbation, we're inducing a plasticity.
Right.
Alvaro, there are many forms of autism. So are you, does this apply to all different children, or is it also specific to the savant form?
No, it's a great question. And we've looked at individuals that don't have a specific gene causing the autism, and we've looked at some that are genetic markers of it, so different types, and it appears that the alteration in plasticity is there in all instances. The specific circuits or the part of the brains that may be affected are different, and how soon or what degree you find is different.
So Nim, you know, we've talked mostly now, we've talked about vision, we've talked about motor system. You know, you study a lot more aspects of emotional phases of development. Mm-hmm. So let's talk about how you've been studying that in these exact same contexts of plasticity and stability.
Mm-hmm. So a lot of our questions are related to the point I made earlier, that we have this design of spending a lot of time with our parents while we're growing up. And so we have been very interested in the role of the parents during this putative, sensitive period for emotional behaviors. And so we know from, you know, since the time of Freud, that there is this very strong association between early caregiving experiences and emotional behavior later on in adulthood. But we don't really know at the level of human brain development why that enduring link exists. And part of the answer may have to do with some of these critical period timing and the influence of the parents. So we've been asking questions about, you know, actually what good is a parent, right? We know that parents are important, but what are they actually doing on a moment-to-moment basis? You know, as I'm rushing my kids out the door to school, shoving breakfast into their mouths to get them out the door, what are those kind of momentary episodes important for? But we also have the opportunity to ask questions about children who experience more forms of adversity early in life. And the reason that we've been very interested in this group of children is because this early adversity exposure is one of the #1 preventable risk factors for emotional difficulties later on in life. And we're just at the beginning stages of understanding why early adversity in particular is so important, maybe more important than later adversity. And part of the answer is related to the concept of critical periods that we're talking about. So you brought up the idea of a double-edged sword before, and I think that there are many double-edged swords when it comes to brain plasticity. And another one of those is that brain plasticity is not really good or bad. It just is, right? It just opens you up to the environment. And so when considering or evaluating brain plasticity, it's also important to look at what's the nature of the environment that the individual is experiencing when that window of opportunity is really open. And so for children that have experienced significant adversity, like caregiving adversity, or what we often call psychosocial trauma, what we have seen, well, two main things. One is that there's incredible individual differences in outcome. So some children show some really significant challenges and others are thriving. So this is an individual differences story that we really aren't very good at explaining right now. But the other piece that we've been seeing, and this is true not only in humans but across a number of species, is that early adversity may actually be affecting brain development on average by affecting the timing of these critical periods. So there may actually be an acceleration of the timing or the opening of these critical periods. So if we know that, that's really important to know in terms of intervention and prevention, because if the timing of these critical periods is shifting as a function of early experience, then that informs how we want to treat individuals and what new experiences we want to give to individuals as a function of these early experiences.
And that's why you like to refer to these, as you said before, as sensitive periods more than critical periods.
For the processes that we're interested in, these emotional behaviors and cognitive behaviors, that seems to make more sense to me because they do seem to be more malleable. And in fact, that's why talk therapy is supposed to work, right? Is that talk therapy is about teaching the brain something new about interpersonal relationships. And the only way that talk therapy can work is if there's continued plasticity in some of these systems.
So we talked a little bit about autism. Let's talk a little bit about schizophrenia, because they're another very complex condition. And there are questions about whether or not schizophrenia may have aspects of a disorder of stability and plasticity. So, Carla, maybe you could enlighten us on that a little bit.
Well, I mean, one main point to make about schizophrenia is that it's now really commonly considered to be a developmental disorder, even though often symptoms appear late, in late adolescence or even beyond. The current idea is that there's this extended developmental critical period, and this period exists beyond the visual system, as Nim has mentioned, and therefore these systems that are maturing much later, including those that have to do with interactions and with cognition and frontal lobe behavior, all mature later. So then the question is, what are the changes that have happened in the brain? And again, it's commonly now thought that this relationship really between plasticity and stability must be perturbed in some way. And when people look into the brain of, not a huge amount has been done, but there's consensus that there are changes in pruning in the brains of schizophrenic patients who have donated their brains for study so that we can actually look at those brains. And in human genetic studies, so looking for susceptibility genes that might produce schizophrenia in children, some of the new gene candidates that have been identified are known in animal models where we study how those genes actually work, are known to regulate pruning in the animal, like the mouse, developmental critical period. So, now you're beginning to hear a kind of common theme that's emerging as we talk, that there are these critical periods that involve a beautiful balance between plasticity and stability, involve pruning, and may have different outcomes, not only through our own experiences, but also when the balance somehow is disrupted by some kind of pathological condition.
So we were saying that, and Alvaro was pointing out that perhaps the autistic brain in some ways is too plastic, and that allows greater performance in some domains. So are we saying here then that potentially a schizophrenic brain is too rigid? Is it overly pruned?
Oh no, I don't think we can say that. I mean, I think there's some suggestion that pruning is excessive. But I think one needs to do many more studies, and it would also be very interesting to have more non-invasive techniques of really assessing plasticity, like Alvaro has been doing in the autistic brains, or other kinds of tests.
So I'm going to throw out something particularly just come to mind. So I have personal experience with this. I have an older brother who's schizophrenic. And we've seen a lot in the news the last few years about how sometimes people who are prone to schizophrenia, drug use, particularly super potent marijuana, more than the average brings it out. And that's my history with an older brother. And so I can't help but think when you're saying this, that when you really tax the brain, whether it's a motor task or pharmacologically, that potentially you're bringing something out because the brain can't recover in the same way that somebody else who didn't have that underlying brain structure function situation. And like I said, I know it's a reach, but I can't help reaching for it.
So we've done some studies with the TMS that I was describing in individuals with at risk for schizophrenia with early onset, early in the course of the disease. One of the challenges of many of these studies is that if you look at human studies on people that are taking medication, the medication, even the exposure to the medication, even if you wash out the medication, will change the way that the brain responds. And so it's really challenging to find patients at a time where they haven't been exposed to medication yet. So as Carla was saying, there isn't a lot of data to support this. But what we find there is almost a reverse effect to what we were talking about in autism, where the effect of the perturbation ends up being too little. It's as if the brain is sort of hypoplastic rather than hyperplastic. But I think more broadly, what we're talking about suggests, to Nin's point, that the double-edged sword at least has two aspects. One is that sometimes the mechanisms of plasticity themselves can be the cause of disease or of symptoms of disease, and sometimes the plasticity mechanisms are okay, but the consequences of the plasticity are disease. You know, we think of it, and you were pointing out in the introduction that it's a mechanism for learning, and it's sort of a good thing. We want to sort of, you know, celebrate plasticity, but sometimes because of plasticity, you develop pain, phantom pain, neuropathic pain, dystonias, alterations that cause great disability because of plasticity. So it's important I think to think of it as something that the brain just sort of has, and we need to understand to the degree of guiding it for the benefit of a given patient or a given person.
But it's interesting too, though. I love it. I love your point, and I think about, you know, if we didn't have plasticity, as Nim pointed out, I mean, we'd all be automatons. You know, we'd all be identical, the same, and the plasticity is really what makes each of us different from each other, in the sense that all of our experiences are stored at the synapses that are being used, and we prune away others. And what this really means is that, you know, if you lost your dog Fido and you decided to get Fido cloned, so you had a complete replica of Fido, Fido would be different because Fido's brain would be different unless you trained Fido exactly the same way and put Fido in exactly the same environment or Fido number 2 that Fido number 1, you know, grew up in. So this idea of individual differences is even present when two individuals are genetically identical.
Absolutely. My dad and my uncle are identical twins, and they couldn't be more different in terms of personality. And there is a lot of twin studies looking at this and showing how the environment, the experience shapes, ultimately the person that they become. And I think it is a celebration of the diversity that we bring, and an opportunity to guide it, and so I think that makes it such a fabulous thing to study, right?
It's funny you say that, because just last night my 15-year-old son was studying cloning for his biology test, and I kept saying, you know, I'd love to have a clone to help me with my work day, but then they'd want my space on the couch to watch the Champions League final in soccer. And he said, Dad, you don't understand anything about cloning.
How did he do on his test?
I think it was postponed, actually. So, you know, we've talked a lot about aspects of this balance. We haven't talked at all about potential for repair and the whole concept about how we could potentially use neuroplasticity to improve and fix problems in a brain. And again, just telling another personal story, you know, 2 weeks ago, I got a call, and I got a call from my dad's wife saying, you know, there's something wrong, he's confused. And the next morning when I could get a hold of him, I talked to him, and immediately I said, you're having trouble with your words, you've had a small stroke, we need to get you evaluated. And the amazing thing was, he knew he was having it, he was having incredible difficulty with expression of his speech, and 2 weeks later now, he carries on a conversation at the age of 86, not normally, but pretty darn close. So obviously the brain's doing it already, the brain's doing it endogenously. So let's talk a little bit about how do we harness recovery of damage after injury. You talked a little bit, Nim, about a traumatic situation, but what about critical periods in childhood? When we get to adulthood, how do we attack that problem? And this is wide open. Anyone?
Well, I think the way we're talking about plasticity really is a huge reason for hope, right? It suggests the fact that our brain can be guided, and the challenge is to learn exactly that, how to guide it. What do we need to do to suppress changes that are these double-edged sword that could cause problems, and to enhance others that can benefit the subject. And we have lots of interesting indications that is possible to do. Ultimately, that's what physical therapy, what occupational therapy, what speech therapy promotes, or talk therapy, is guide the changes for the benefit of the subject. But I think we have now, as we understand more of the molecular basis and the physiologic basis of these mechanisms, we have ways to target this mechanisms with medications or with devices, with brain stimulation that more directly to the brain promote the changes. And that's happening, and it's happening guided by basic research and translating that basic research to humans.
Yeah.
Sorry.
Yeah, one of the things that I'm really excited about in terms of intervention is thinking about how can we boost the power of current interventions by knowledge about critical periods and plasticity. So if we can increase plasticity prior to some known effective intervention, that's really powerful. And so, as you mentioned, there are many examples in basic neuroscience where pharmacologic or genetic administration or modification can actually increase plasticity and in many ways bring the brain back to that childlike state to allow for some environment to have a really big effect on the brain. That's really powerful, and there's a demonstration in humans too that has actually used one of these pharmacologic manipulations with people who don't have perfect pitch, and perfect pitch depends on critical period learning early in development. So perfect pitch was able to be developed in adults who had taken this pharmacologic drug. So then the question is, well, why don't we all do that? Right? And the authors of the study they're very careful to say that, hey, remember, the brain actually spends a lot of energy to prevent these critical periods from opening up again, right? The brain wants efficiency. The brain wants reliability. And do we want to remove all of the lessons that we've learned throughout our entire developmental history? Do we want to remove aspects of our personality and so on? So it becomes a very tricky question, even though there's a lot of promise for recovery following any type of trauma.
But in that perfect pitch study, they didn't, the people didn't lose. They learned, right, something new. So there's hope, really. There's hope for having selective, and a selective intervention. So I think that's exciting. I mean, from the animal studies, so from looking again at the mice, and actually one of the authors of that study also has done work looking at the mouse critical periods for vision, as has my own lab. And what we find is that you can actually go into the adult mouse brain and really restore juvenile-like plasticity in the adult brain. And the mechanism of doing that seems to be, again, to allow for new synaptic connections to form. And if we study the story of the childhood cataract, but now in the mouse, then remember I told you that if children have a cataract and it's not immediately operated and clear optics restored, then they become either permanently blind or seriously, vision is seriously diminished in that eye. And really, there isn't a good way of treating that blindness. It's called amblyopia in adulthood. But in these mice, one can actually make the cataract model and then go in the adult mouse and engaging these childhood mechanisms of plasticity again, affect recovery of vision in the eye that was blind, which is amazing. And it's really demonstrates, I think, that by understanding the underlying mechanisms that regulate these developmental critical periods, it's possible to go in and then to manipulate them again. And to me, it demonstrates another thing, which is that there must be in the adult brain, and presumably in our brains too, because we have many of the same molecules that are being studied in the animal models, there must be brakes on plasticity, because these manipulations essentially take the brakes off. And then, of course, you can worry about stability. So then we come back to this whole theme again. But the idea that there's actually more plasticity in our adult brain that we can tap into, to me, is really exciting, because you could say that even if there are some downsides, that after a stroke or damage to the brain, wouldn't it be wonderful if it were possible then to tap into that latent extra plasticity even briefly and combine it with training in physical therapy? So somehow a pill that would let me speak French without an accent could actually have a really important therapeutic value, which would be to really help people recovery from brain damage.
Yeah, so if we look here, sorry, go ahead.
No, go ahead.
Well, I was just going to say, what we have here is a video of a modern rehabilitation technique with a split treadmill. Maybe, Alvaro, you can tell us about the concept behind this.
Yeah, so part of what physical therapy interventions like this, or like something called constraint therapy that some of you may have heard about, in individuals that have stroke that cannot use both legs in walking correctly or cannot use one hand correctly, we've come to learn that if you force the use of the other hand, of the impaired hand, by limiting the default of just using the unimpaired hand, or by, in this case of the video, if you force the walking correctly by aligning the paretic leg, because it cannot cross over, and so therefore limit the pattern of abnormality that normally would happen, that because of this repeated training doing the right thing, that promotes the rewiring, that promotes the plastic change and speeds up the recovery. So you can, by guiding, by constraining the behavior, use that as a way to improve the recovery process.
But
the Holy Grail, I'm sorry.
But it has to be done very specifically, right?
Very carefully and specifically.
Right, because if you learn bad habits during that period of recovery, then the recovery is going to be much less effective, right?
That's right. So it's not about doing a lot of movements despite the fact that they're impaired, but actually doing very carefully controlled movements the right way, which of course is hard to do because of the problem that the patient has. And so the opportunity of opening up the mechanisms of plasticity in such a way that they become more active and therefore benefit from the intervention more becomes really appealing. And examples of that do exist even in humans. You know, not only with medications. One of the things that we know about Alzheimer's dementia, Alzheimer's disease, is that before the clumps of this abnormal protein come together and deposit the plaques in the brain, they are floating around, and in that stage already they alter and damage synapses. So they become damaging substances for the mechanisms of plasticity, as it were. If you take patients with Alzheimer's disease and you try to get them to learn from video games or computer games and get better at cognitive tasks, they derive very little benefit. It would take too long to get a benefit that is sort of meaningful. But if you activate that circuit with brain stimulation, for example, just before they actually engage in the cognitive training, now you get more benefit from the cognitive training. So combining interventions, device-based or pharmacologic interventions with behavioral interventions, I think is really a way to leverage what we know about the mechanisms of plasticity in a way that translates into benefit for patients.
So it's going to be a combination of understanding, as you've all been saying, it's not an open or closed door, but how do we change the degree of that, but in a very specific way that's very specific to the problem, whether it be a childhood trauma, a stroke, Alzheimer's, something else, any of those sorts of lines. Because it's interesting, you know, Carla, you also have a history of stroke that led you to some degree into neuroscience, correct?
Oh yeah, well my paternal grandmother, who was a great tennis player and the first person in our whole family to go to college, had a really devastating stroke that left her completely paralyzed on the left side. And I had two uncles who were both neurologists, and this was many, many years ago when I was just in college. And they were excellent at diagnosing where the stroke was, but could not help her at all. So that really influenced me to become a neuroscientist as opposed to going to med school. I mean, it's different now, you can do more, but we could do more even in the future, I'm hoping. But I realized we needed to know more about what was going on in the brain. And you know, to that effect, I'm looking at you as the neurosurgeon, and you probably know better than everyone that when the brain is damaged, or when you have to remove a part of the brain because of either epilepsy or cancer, the brain neurons do not regenerate themselves. And so really what's left is the plasticity in the remaining circuits that really is the hope for tapping into or retraining or stimulating or whatever.
So let's take that a step further. You know, one of the things that I'm always curious about in that exact concept is that in the operating room, we have a pretty good idea of what's going to happen. We have a pretty good idea of what area of the brain we're going to go into and what consequence it may cause, but we spent very little time saying, how do we take these concepts of plasticity and apply them prior to an insult? Or for, like, Nim, if you knew that a child or a young adult is going to go off into some traumatic environment, let's talk about ways we could envision harnessing these concepts for not rehabilitation, but prehabilitation, treating prior to an insult. And is this realistic? Is it a pipe dream?
I think it's totally realistic. I think that's where things are going to go. Is if you're able to shift the organization of a given function in the brain so that, say, language areas from other parts of the brain contribute to language in a more meaningful way, so that when you go and do the surgery, you can take more of that left hemisphere out, which means you can remove more of the tumor, and therefore the life expectancy is better. That is actually possible to do. You can use brain stimulation to suppress parts of the language circuit, effectively forcing other parts of the brain to take over speech.
So you're lesioning before a lesion.
You're virtually lesioning. You're cooling it down. You're sort of making it hard for the brain to use those resources, forcing it to find other resources. You're not lesioning it, but you're putting brakes on it. And I'm sure with more understanding, there would be even better specific brakes, even at the molecular level, that one can use. But the idea is, if you make it hard to use what comes easy, just like William James was anticipating with behavior, will the brain find ways to maintain the function by reorganizing? And remarkably, it appears that's exactly what happens. And if after 6 weeks of forcing that system to change, you do a language mapping, and more of the right side or more of the posterior parts of the brain are contributing to language and less of the frontal areas, and the surgery can be done with a larger resection.
So Nim, we're talking about, language is easy to test and identify, and the motor system is easy to test. When we get to much more complex things like social development and emotions, how do you even think about tackling that problem? Because there are situations where that children are going to be undergoing a stress. How do you even take that on?
I think there are pluses and minuses, in that problem. So the challenge is that these social and emotional behaviors are, including the whole brain, right? It's not a single hotspot that is doing that behavior. It's really a distributed process that's engaging so much of the brain. So it becomes really hard to localize in the way that we've been talking about for things like vision, for example. On the other hand, I also think that opens up the potential for intervention for someone who's experiencing challenges, because if you're getting a lot of systems feeding into this very complex behavior, then that gives you a lot more opportunities for tweaking some of these nodes in order to improve, for example, the ability to regulate your emotions.
So we've talked a lot about aspects of disease and repair. What about this holy grail idea? What about the idea of actually enhancing the brain? Whether one day, I was thinking today, I had a long day, and I was thinking, oh my gosh, I got to focus on what we're doing tonight. And I jump in a car, and when's the helmet going to come out through the ceiling or already been built into the roof, where I can say, if I can just get my temporal lobes going and my memory working a little better, like how great that would be. Like, how far off are we? How far off are we from everyday enhancement of function?
It didn't work this morning.
Exactly. You were in the wrong car.
I was in the wrong car. Well, so, what gave me pause is that, of course, there is a lot of industry investment and really direct-to-consumer devices already claiming that that's possible, that we don't need to wait at all, that you can just do it today. You can build, your own brain stimulator devices and build it into your car if you want to, and people will claim and try to convince us that it is possible to enhance capacities. The problem with a lot of those data is that what they show is that it's possible to get better at performing a given task, not to actually get better at the fundamental skill that underlies the task. So I think that the attempt to get us there is already here, but we need greater understanding, including greater understanding of that double-edged aspect that Nin was mentioning. If we get better at one thing, will it be without a cost, or will the brain allow you to improve something at the cost of something else? So, for example, you can ask a question and say, what if we were able to get better at paying attention to this part of the world? We know a lot of what the circuitry is that allows us to pay attention to this particular spot. And so you can actually identify those brain areas in humans, apply simulation that increases activity in those brain areas, and you can make people get better at attending to this part of the world. But when you do that, it comes at the cost of attending to this part of the world. In fact, it does so because these two areas sort of are almost at a yin-yang kind of a relationship, and so there are interactions between the two hemispheres, and so if you improve one, you worsen the other. That's easy to test in something as concrete as spatial attention for a visual task, in something as complex as overall behavior or more higher order cognitive functions. What exactly the cost might be is much, much harder to test. And so I think it raises not just methodological and biological questions, but real ethical questions. If you want to improve certain skills, what would potentially be the downside of it?
Well, I mean, so let's back off a little. I mean, humans are complicated. We've got big brains. You fix one thing, you complain about something else. But mice, you can take it apart in mice, right, Carla? So what happens there? What if you make mice more plastic?
Well, you can, and there are a number of examples where these break genes have been taken off, and for plasticity. And usually it's in order to study the function of those genes during these developmental critical periods. But if you then look at what this does to the whole mouse in terms of its overall behavior. So now let's go beyond the visual system and just ask, okay, when we remove the brakes and we can open up the critical period again in these mice, how do they behave? Are they smarter? Are they dumber? And in some cases, the mice that don't have some of these brake genes are performing at much higher levels than normal wild-type mice. Now, when we study mice in the lab, we're not really looking at their full repertoire of behaviors. So we can't test everything. But it's pretty amazing that they can perform at higher levels. And what do I mean by that? I mean that mice can learn wonderful memory tasks. So they can run through a maze and they can remember the whole, the turns in the maze and so on. And these brakeless mice actually are able to learn and remember that task much better than mice that have all 30,000 of their genes intact, for example. And so far, it's not clear what the downside would be, but we've only tested them in pitiful, simple ways, and it's not the full repertoire even of mouse behavior. So I go back to Alvaro's point, which is that there may really be serious downsides. Now, that may not stop a student the night before an exam for wanting to take, whatever good pill might work in the future. And I agree with you. I don't think we're not that far away from having some much more mechanistically informed ways of manipulating or increasing brain plasticity, whether it's from stimulation or from pharmacological approaches.
So let's ask that question then. I mean, we have aspects that all of you work on that lend themselves to pharmacologic, lend themselves to cognitive behavioral therapy, yet lend themselves to interfere or enhance the brain with stimulation of networks and circuits. So is it drugs? Is it electricity? Is it behavioral therapy? Is it all of the above? Do you need a cocktail? I mean, how do you guys see this playing out?
Well, Alvaro, you were saying that really combining seems to be very effective. In other words, if you just use TMS, maybe not as good or even at all as if you pair the TMS with the actual task that you're trying to work on. And I think the same thing is going to be true with pharmacology, with drugs. The idea again would be you don't just take a drug, but maybe before that exam the night before you actually rehearse everything beforehand, right? So you really
You're
have
still
to.
going to have to take French lessons.
Okay, good point. And
my 15-year-old son's still going to have to study cloning.
Yeah, exactly.
Right. Exactly.
Right.
You can't just take the pill. But it's fascinating when you think about it, right? Because it's all about, we said it already, but it's about pairing, right? I think we have a couple of images of deep sea divers in Burma who humans, when they have to go into extreme conditions, the divers, as they go down, they actually focus so well that they change their eye musculature and the shapes of their cornea so they can be able to focus at depths that normal humans cannot. And so already pushed to extreme, the human brain has this remarkable capacity. And so you have to think that whatever this is in terms of potentially enhancing function, it's got to be paired incredibly specifically in how it's done.
Yeah, and I think it has to ultimately be really personalized. I mean, that's to me one of the beauties of this type of insights that we're gaining, right? That is going to be relevant how you pair it and what you pair, but also where you start from. And that is going to be different for every single individual. And so the kind of exercises, interventions that lead to the best result in me are going to be different, even for the same thing, different than the ones that work in Nin. And that there is real personalization in that, real unique targeting, which I think is going to be a lot more impactful, whether it's enhancement or whether it is recovery from function.
So what about memory? I mean, memory is something, to varying degrees. We're all here slowly moving along chronologically, and everybody worries about what's going to happen to your memory. And in my world, people have the ability to sometimes put electrodes into the brain that allows us to stimulate the brain. And there's a number of reports of different parts of the brain. If you stimulate it just the right way, you can make somebody's memory performance better. And how do you think of that as not a way to combat aging per se, but to help people adjust as they get older and keeping memory intact? Forget about super memory, even just trying to keep memory intact as we go along. How do you guys see us approaching that?
I mean, correct me if I'm wrong, but aren't there less sexy, innovative ways that people think about or have studied to enhance some of these processes? So for example, the things that we know we should do, like getting good sleep, exercising, engaging in cognitive stimulation throughout a lifetime, have been associated with better cognitive aging in the long run. So those are hard things that we all know we should do, but routine things that we should be doing that may actually be improving some of this retention over the long term.
So lifestyle modifications do work, and do work through plasticity. So physical exercise promotes cognitive function. I mean, and in fact, if you do an analysis of all the studies that have been done in the literature, which we just published, there is I don't know why this is the number, but there is about 52 hours a day. 52 hours, talk about enhancement.
Only in my day.
It's really hard work.
It's really hard work. Yeah. 52 hours in 6 months, actually. Yeah. So it's not a lot. And it doesn't matter how many hours per day you do. It's sort of a total amount of dose, almost. But you need to put in some hours, but it's not a ton of hours.
Of exercise?
Of physical exercise that improves cognitive function in the elderly. So I think that there is an effect of physical exercise onto the brain, and it appears to be linked to mechanisms of plasticity, in at least as one can probe them in humans with brain imaging and brain stimulation techniques. But I think rather than getting at the level of suggestions, you should sleep better, avoid medications that are bad for you, eat the right thing in the right amount, and exercise and challenge your brain with new things and have social relationships. All those things are true, but we should be able to get to enough understanding that we can prescribe it, that we can tell Nin, this is the number of hours of this exercise that you need to do, and here's how we're going to help you actually convert these hopes into realities, right? Because it's hard to stick with a program like that. So I think that's the step that we still don't have, is the understanding of how to actually make this a prescribable intervention, like we do with medications. We don't tell patients, take something for that. We tell them what to take and how much and so forth. And so I think we need that level of understanding.
So, but yet it does, like you say, exercise, everyone agrees is good, eating well, but it's got to be specific too, right? Because you talk about if somebody's a really good tennis player and they start playing squash, it's a racket, it's a ball, but they say that squash tears apart their tennis game. And if somebody's trying to learn 5 languages at once, as a child, they can learn them all without an accent, but the time they acquire any one of them is delayed relative to a person learning 1 or 2 languages. So it's interesting because we also have to understand that aspect of things about, about what's good globally versus what's good locally. So, but what about happiness, set points? right? Everybody wants wellness. So is it possible that we're going to someday understand the brain well enough to say, okay, you've been through a really terrible life trauma, but we can actually help your set point?
Well, I think, I would think about sort of maybe not happiness, but that feeling of being able to regulate your emotions, to be able to feel less sad when you want, to be able to feel more sad when you want, to kind of have that emotion regulation ability. And so there are studies, for example, with people who meditate, which we generally think of as a healthy lifestyle practice. But there are different levels of expertise in meditation, right? So there are Buddhist monks who do this for, say, 40,000 hours in total versus really, really dedicated meditators who might have done this for 20,000 hours, and then novices. And this research has shown that the amount of meditation practice being the key word, that the behavior has to be routinely engaged with, is associated with brain changes in the regions of the brain, like the prefrontal cortex, that we know are associated with emotion regulation. What's interesting in that finding, and it relates back to the original walking example and the quote about habit, is that there was this inverted U-shaped dose-response function, such that the individuals who were meditating a lot, but not to the degree of the Buddhist monks, actually had the most different prefrontal cortex activity, and then it came back down again for the Buddhist monks, suggesting that there is this element of plasticity in the brain regions that we know are associated with emotion regulation, but an element of habit can come into play as well, so that it starts to become more of a naturally occurring state of the brain that can be, as you were referring to before, kind of the core process that's being affected rather than the more superficial.
So I would have to say in summary that obviously we have a really, really tight balance between plasticity, stability, but as we've all learned, there are incredibly exciting things, not just normal behavior, attacking diseases going down the road, but in the meantime, eat well, sleep well, meditate once in a while, exercise a whole lot, and surround yourself with really smart people who make you happy. And for that, I thank all of you.